Thanks, Ove. It is a complex title, I'll acknowledge that, but it's for a complex organism, and one of my goals today is to convince you that corals are not simple organisms, as is commonly thought, nor are they merely fish habitat, as many of my good colleagues sometimes would have you believe. Corals are complex partnerships, and each one of these coral species, we know that they're morphologically diverse, but more than this, they actually represent a complex association between organisms from at least 3 different kingdoms. And the main players in these partnerships are the coral animal, photosynthesizing symbiodinium or dinoflagellates that live in the coral tissues, and bacteria that live in the surface mucus layer on the surface of corals. Also, we know that from some corals there have been more than 30,000 types of viruses isolated, so it's quite likely that viruses and archaea are also players in these partnerships. And the combination of all of these organisms creates what we call the coral holobiont. Now, Ove has already elegantly laid out some of the issues that are facing corals now and in the near future. What we'd like to consider are some of the options for the coral holobiont to actually respond to climate change. But before we can do that, we need to understand how each member contributes to holobiont health and how the environment and changing environments affect each of these partnerships. For a very long time, almost 4 decades now, people have been researching the partnership between the coral host and symbiodinium. And we know that this is a very important partnership, contributing nutrients that aid in the growth of the coral. And also we've shown more recently that it can have impacts on the thermal tolerance of the holobiont. And if that partnership breaks down, of course, there are dire consequences with bleaching. More recently, people have been researching the relationship between the coral host and many of these associated bacterial communities. And we know that bacteria produce antimicrobial compounds and other secondary metabolites, as well as assimilating trace nutrients. And when these partnerships break down, the coral becomes more susceptible to pathogens, and we often see disease. But in fact, there hasn't been a lot of work done looking at the relationship between the symbiodinium partner and the associated bacterial communities. And that's something that our group has been working on for the last couple of years and will be the focus of some of the things that I'd like to show you today. We know one of the paradoxes in coral reef science is that coral reefs do grow in nutrient-poor waters. The discovery of the tight cycling of carbon between the coral host and its symbiodinium has answered that paradox to a certain extent for carbon. But what about nitrogen? We know that coral reef waters are certainly nitrogen limited, and moreover, only bacteria that contain the nitrogenase enzyme actually can fix nitrogen and convert nitrogen from a gaseous form into a biologically usable compound. And recently it's actually been discovered that there are nitrogen-fixing bacteria that are associated with corals. And so one of the students working with David Bourne at Ames and myself, one of her goals has been to compare some of these nitrogen-fixing bacterial communities among different coral species, so in fact 3 different coral species, at 3 locations to try and get a handle on whether we are seeing very stable nitrogen-fixing communities associated with corals, which might suggest that there actually there is tight cycling between bacterial communities and symbiodinium within a coral species. This shows you a PCA of some of the clone library sequences for the NIFH gene, and I think what becomes clear is that each of these the clone libraries for a given species, in this case Polylepidemicornis, down here in Acropora, and over here another Acropora, the bacterial communities are certainly clustering at the level of species and not location. So in each one of these circles, you've got the 3 different reefs. So it looks like, yes, nitrogen-fixing bacterial communities are stable and species-specific, which would suggest that they are highly co-evolved. And that there are stable relationships between the coral bacterial communities and, in fact, with implications for the Symbiodinium. This is a neighbor joining tree showing the phylogeny of the NIFH gene, and one thing that is quite startling is that about 40% of the sequences are actually from the Alphaproteobacteria. These are two very abundant nitrogen-fixing groups, which actually have 98% similarity to the Rhizobia group of bacteria. And these are a very important group of nitrogen-fixing bacteria that are associated with legumes. They live in the root nodules, and they provide the plant with nitrogen. And in fact, it's such a highly co-evolved symbiosis that they actually can't fix nitrogen when they're outside their plant host. So I guess the question arises, is there a similar type cycling of nitrogen fixation and the Symbiodinium within corals, and is the relationship that highly co-evolved? And if it is, it also calls into question a commonly held assumption, and that is that nitrogen limitation in the seas is actually regulating Symbiodinium populations within the coral host. So again, we still have a ways to go to really understand those relationships. Next, I'd like to have a look at another important element in marine systems, and that's sulfur. Sulfur, in fact, corals are the largest producers of two sulfur-based compounds, DMSP and DMS. In fact, produce more than an order of magnitude of these compounds than any other organism on the planet. We know that DMS aerosols actually are important in enhancing cloud formation. So in fact, DMSP produced in the oceans and through bacterial processes broken down to DMS actually is functional in promoting clouds, which of course deflect solar radiation and reduce surface seawater temperature. So there is a nice cycling that goes on in the sulfur cycle. And in fact, it's the Symbiodinium within corals that do produce the DMSP. If you look at a coral, and these are all these little golden dots are actually zooxanthellae in the coral's tissues. They actually occur at concentrations that would be equivalent to a phytoplankton bloom in the oceans. So enormous concentrations of Symbiodinium within coral tissues. Now we know, as I said, that bacteria cleave the DMSP to DMS, and interestingly, DMS has very important reactive oxygen species scavenging properties. So it looks like DMS may potentially be an important antioxidant for corals. So as well as having a role in cooling local weather and regional weather, DMS and DMSP production may actually have a role for coral-associated bacteria as an antioxidant. So as a consequence, another one of David and my students, JB, has actually done a fairly extensive literature search and found that looking at all the bacteria that are known to metabolize DMSP, and this is a list of those bacteria. This is a list of bacteria that are also able to metabolize DMS, and all of these ones highlighted here in this interior blue circle are bacteria that have been isolated from corals and therefore potentially have a role to play in terms of DMSP metabolism. So corals do harbor many bacterial strains that are able to metabolize these two sulfur compounds. He's then gone on to isolate, identify, and actually quantify some of the coral-associated bacteria that are able to metabolize these two compounds. He's done this for 2 coral species and looked at both at mucus, tissue, and skeleton samples on a variety of media, and using just either DMSP or DMS as the sole sulfur source. And here, this shows the clone libraries for all bacteria that he found in this study. If you then look at, focus just on the Alphaproteobacteria here and the Gamma-Proteobacteria, which are shown here in the gold and the brown columns here, and this is being shown for mucus samples and tissue samples from these 2 species, what becomes very clear is that the percent of clone libraries that these 2 different bacterial groups comprise is quite large. taking that one step further, these diagonal lines now show you the percentage of those two groups that are actually able to metabolize those sulfur compounds. This is according to the study that he did. So, looking at these percentages, it's clear that a major proportion of bacteria found on corals out of the whole clone libraries are able to metabolize these two sulfur compounds. And in fact, he isolated and identified several new sulfur or DMSP and DMS metabolizing strains from both of these species. Quite exciting. Going back to this diagram, and just to briefly recapitulate, I've shown you here that bacteria are fixing nitrogens, which have implications for the symbiodinium partner. And I've just shown you that the symbiodinium is producing large quantities of DMSP, which actually has forms a substrate for the bacterial communities within the coral. So it looks like these bacterial and symbiodinium associations are quite prevalent within the coral holobiont. What's more, bleaching and the consequent potential loss of antioxidants from the breakdown of the DMSP to DMS would actually amplify the physiological stress associated with the breakdown of the symbiodinium-coral relationship. And so certainly these bacterial symbiodinium symbioses look like being very integral to coral health as well. Now, to carry on, how does thermal stress, though, affect this level of the symbiosis? So how does thermal stress affect the relationships between symbiodinium and bacteria? And next, then, what I'd like to show you is another study, and this time using a system that we've been able to use to manipulate the symbiodinium associated with corals. It's quite a nice system where we can spawn corals, collect the gametes, fertilize them, settle the larvae on tiles so that you end up with small little juvenile corals growing all over the surface of these tiles. And then we can experimentally inoculate them with either with several different types of symbiodinium strains. So in this case, in this study, either with symbiodinium D or C1. And then grow the juveniles out, and in this study, for 1 year at Magnetic Island. And this was a study then by another of David and my students, Rachel Lippmann. And she showed that, in her study, she actually took these juveniles that had been infected with C1 or D symbiodinium, grew them up, the juveniles, or exposed them to 2 different temperature treatments, sort of an ambient temperature treatment and an elevated temperature treatment, and then followed the bacterial communities through time. Just a brief note here that when they were retrieved from the site at Magnetic Island, in fact, they were starting out in an ambient temperature of 30 degrees, so they were brought back into the lab, put at 28 and 32, and in fact, already at that point, we'd lost some of the juveniles associated with symbiodinium D because of mortality. She looked at the coral-associated bacterial communities using both clone libraries and DGGE of the 16S RNA gene. And also she confirmed that the symbiodinium clade was actually the same that we had inoculated them with using the ITS1 marker. And this shows you a PCA of the dominant bacterial types. And basically, I just want to draw your attention to 2 points in this PCA. This shows you that the group of juveniles that were associated with the clade C1 all clustered together regardless of the temperature treatment. So regardless of whether they had been ambient at time 0 at 30 degrees, taken to the 28-degree temperature treatment, or up to 32. In contrast, those juveniles that associated with clade D, yes, they clustered with the ones at, that had clade C1 here at the beginning, or in other words, when they were put in the 28-degree temperature treatment, but otherwise they were basically all over the place. On day 1 of the treatment, they had a lot of E. coli in the clone libraries, but by day 12, in fact, the bacterial communities were largely driven by the presence of Vibrios. So we saw quite a dramatic difference in the bacterial communities associated on the same coral species, but driven by the two different Symbiodinium clades. So just to highlight this in the clone libraries, Symbiodinium C1 juveniles, they had very low percentages, well, in fact, no Vibrio to start, and no at 28 and very few at 32 degrees, whereas juveniles associated with Symbiodinium D in the field had 13% Vibrios. When they were put in a cooler treatment, that number reduced. But at 32 degrees, more than a third of the clone libraries were comprised of the Vibrio species. The DGE profiles confirm this. There were very distinct differences in these bacterial communities. This very prominent band affiliates with Vibrio corallilyticus, which we do know can cause some types of white syndromes. And again, just looking at the Vibrio profiles, there were many more Vibrios in the juveniles associated with clade D than there were in the ones associated with clade C. So, yes, when we do thermally stress this particular association, we do see that the Symbiodinium type actually seems to determine the stability of the bacterial community to temperature stress, and those that are associated with Symbiodinium D are more susceptible to Vibrios. So even though Symbiodinium D is often characterized as more thermally tolerant, there may be a trade-off going on there where they're actually more susceptible to pathogens. What other options are for the coral holobiont in terms of adaptation to thermal stress here? What I want to do very quickly now, I'm living in fear of the bell, is show you a very neat study done by Emily Howls, who's a student of Madeline Van Open and myself. She, again, manipulated the Symbiodinium populations, but this time she used the same type, Symbiodinium C1, but just from two different populations. And here, this shows you Symbiodinium source came from Magnetic Island, which is a warmer population, and Whitsundays, which is typically a 2-degree cooler population. We know there's limited gene flow between these two populations from previous work. She did the same. She was able to rear the juveniles in controlled conditions, infect them with now two different populations, that warm-adapted and cold-adapted populations of the one type, Symbiodinium C1. She inoculated then the juveniles with, as I said, either Symbiodinium C1 or D. She then subjected them to a heat stress experiment and looked at photochemical efficiency, and you can see very clear differences. Increased photochemical efficiency when the corals were associated with the C1 from magnetic populations, decreased from Whitsunday populations. Very quickly, very clear differences in cell density of the Symbiodinium. The ones associated with the cooler populations actually bleached at 32 degrees, whereas the ones associated with the magnetic island populations, you didn't see any difference in cell densities. This also had effect on the coral host, so the growth of the coral host increased when it was associated with C1, whereas when it was associated with, from Maggie Island, when it was associated with C1 from the Whitsundays, the growth decreased at higher temperatures, and you can see that there. So, just to summarize briefly, Symbiodinium populations, it turns out, are actually adapted to the local thermal regimes we can see that from the photochemical efficiency, growth, and bleaching data. And that local adaptation has an impact on coral fitness, as we can see in terms of growth or partial mortality. So it does look like there is thermal tolerance variation in the Symbiodinium, both at the clade level and at the population level, which gives increased options for the holobiont. We're still unraveling the enormous complexity of these coral microbial partnerships, but it does appear that there is some further potential given this evidence of local thermal adaptation. There are still many unknowns, and I guess I'll take my lead from Graham Farquhar's comment this morning. It's my fear that even though we do have some evidence, John Pandolfi showed us evidence of corals are in decline, but it is my fear that we're actually learning about these very fascinating creatures in the twilight of their years. Thank you. you.